I am pleased to introduce tonight's speaker, Dr. Folami Ideraabdullah. I messed that up after all my practice. Have any of you been practicing how to say her name right along with me? She is here to present the second of the 2013 Appetite for Life Academy lectures. Dr. Ideraabdullah is the newest member of the NRI's research team, and she joined the NRI last month as an assistant professor of genetics, and she holds a joint appointment with the School of Medicine at UNC Chapel Hill. Her long-term research goal is to understand the role of gene-environment interactions in phenotypic variation and disease. That's basically genetics. Dr. Ideraabdullah earned her BS in biology at Pennsylvania State University and her Ph.D. in genetics and molecular biology at UNC Chapel Hill. An accomplished scientist, her prolific contributions as a graduate student earned her a Sarah Graham Keenan Edwards Hobgood Dissertation Fellowship. She continued her training as a postdoc at the University of Pennsylvania, where she was awarded a Ruth L. Kirstein National Research Service Award from the National Institute of General Medical Sciences. Dr. Ideraabdullah was born in the U.S. She grew up in Liberia and moved back to the U.S. at the age of 15. She knew since she was a little girl that she wanted to be a scientist. She said that biology fascinated her in the 7th grade and genetics fascinated her as a 9th grader, and that fascination led to her life's work. We are pleased to have her with us tonight to tell us about her work investigating the relationship between diet, genetics, and the environment, and how this research can give each of us a chance to live a healthier, fuller life. Thank you for joining us this evening, Dr. Ideraabdullah.
All right, good evening. Can everyone hear me okay? Or do I need to turn it up? Everyone can hear me. All right. I just want to thank her for that wonderful introduction. That's probably the best introduction I've ever had. It's very thorough and actually accurate. And so I'm really pleased to be here tonight and to be invited tonight. So thank you to those who are organizing behind the scenes. But I'm really pleased to be here tonight because this is an opportunity for us to tell our community what we're doing and talk about ways in which you can be involved, right? So I'm going to try to save questions until afterwards, but if you have a burning question that you want to ask in between the talk, just raise your hand and I hope I see it. So I hope this talk is informative. I hope you get an idea of the kinds of things that I'm working on here at the Nutrition Research Institute and the kinds of questions that scientists are asking right now in this field of nutrition, genetics, and environment. And so today I'm going to talk about the heritable effects of diet. And I titled this talk, Becoming What Our Parents Eat, because that's how I think of heritability, and that's how I think of diet. I think of what kinds of things do I eat that could potentially affect my children or my children's children and their children, and so on and so on. And so I want to start off with this slide because I think you are what you eat is a very common phrase. Everyone's kind of heard it, meaning if you eat well, your health will be well. If you eat poorly, your health will be poorly. Roughly said, that's kind of what it means. And I was surprised to find that it was coined by a French gastronomer in 1826. And actually what he said was, tell me what you eat and I will tell you what you are. And I also wanted to put this picture of this vegetable fruit flower man up there that was painted by a very famous Italian painter. And I believe it's displayed at the Louvre, but I'm not sure, to be honest, where it is now. But I put it up there because it reminded me of a picture that we had in my house when I was growing up of what I call the vegetable man. I thought he was creepy at the time, but now I understand that the vegetables you eat and the fruits you eat and whatever you eat or put into your body really determines what your body becomes. Maybe you won't have a fruit or a grape on your head, but it does determine what your body becomes, right? And so let's talk a little bit about what our parents eat, because what he was talking about is what the individual eats and what they become. And so what I'm going to talk about today is what we eat and how that affects our children and children's children. And I put this picture up here because of course, what the mother eats while she's pregnant plays a major role in what her child becomes. And I think sometimes she gets, an unfair or unequal rap for, having to eat healthy, whether it's a salad or, a fat, juicy burger. Sorry for those of you who are hungry tonight. But I think also we have to think about what our fathers eat as well, because there's new studies showing that father's diet may play a role in what the child becomes, and that's whether he's drinking his child's breast milk or having some Jack. All right? So I just wanted to point out that, I'm going to talk a little bit about some studies that talk about the effects of what our parents eat and how that contributes to what we become from a child to an adult, right? So we're talking about early origins of disease. So even though you may realize you have a disorder later on in life as a mature adult, this disorder might have started from effects that occurred in utero when you were still in the womb. So we're talking about early origins of disease and how these types of effects that you can get early on in development can affect your health and fitness outcomes later on or throughout development. And so in order to talk about these things, how our parents' diet affect us, we have to talk about heritability. I'm sure most of you, if not all of you, know what being heritable is, but I'm going to point it out anyway for those of us who don't. So heritable means capable of being passed down from one generation to the next. So here we have a nice, I guess I don't have a pointer, but here we have a nice square-bodied woman with beautiful orange hair, and her husband or her mate is a square-bodied man with blue hair. Their child comes out with a square body and nice orange conical hair, right? So the idea here is that he's inherited his square body from his dad and his orange hair from his mom. And the interesting thing about things that are heritable is that they can be passed through multiple generations. So let's say this little guy grows up, he meets a nice round-bodied woman with green hair, and they have a child. And their child inherits, the square body from his dad and the orange hair from his mom. And what you can see with this pedigree, which is what it's called, is that he inherited his square body all the way from his grandfather, and he inherited his orange hair all the way from his grandmother. And so the point here is just to think about some of the things that we do during our lives and how they can affect multiple generations potentially. And so how are these types of traits are inherited? How do we get our square body and our orange hair in this case? And so obviously these comes from parental contributions, right? And we're not going to talk too much about this because I think we're all mature adults in here. But the mother contributes an oocyte or an egg, and the father contributes sperm to this offspring. And these are two individual cells, but the important things about these cells and what they contribute to the child is that they contribute genetic material, right? And so this is where the genetic material that makes up this child comes from. The human genome we all in each cell in our body have 46 chromosomes, 23 from each parent, right? So 23 from mom and 23 from dad. And these chromosomes all have little bits on them called genes. And there's estimated about 20,000 or more genes, and it depends on what you consider a gene and how you define it. But the literature says about 20,000 plus protein coding genes. And what these genes do is they establish a pattern of activity. So they're either on or off, right? So in this case, gene A is on or off, and this is what's going on in our cells during development. Now, along a chromosome, you have multiple genes, right? And you can see there on the bottom. So this is gene A, B, and C, 3 genes. And along your chromosome, you have genes sort of in a row, right? And some of them are on or some of them are off. So these are patterns of gene activity, or what we call patterns of gene expression. And that's what determines whether this little guy inherits a square body or orange hair. Okay? I hope that makes sense. Now, there are a lot of DNA sequence differences at these genes or in your genome on these chromosomes that I mentioned earlier. Most people would say that humans are about 99.9% genetically identical, but there are some differences, right? So in this case, I'm showing my little blue man here has a single change in his DNA sequence. So those A's, C's, G's, and T's make up our DNA sequence. And he's got a single change, which I've indicated with this red arrow. And the little green guy has a different DNA sequence at that location in his genome, which is a G. Okay? And so this genetic difference actually, in some cases, depending on where it is in your genome, can directly affect whether your gene is on or off. And so if this is in a regulatory region, in this case, you can have gene expression differences between these two men. In one case, gene A is on, in the other case, gene A is off. And that could directly contribute to whether you have a little blue person or a little green person. We're thinking very abstractly tonight. All right. And so that said, these genetic differences are very important for the outcomes or the traits or the healthy outcomes that happen to you during development or that develop during development. Now, scientists have decided or concluded based on looking at the different types of traits that are out there, the different types of diseases that are out there, et cetera, that 0.1 or 0.01% of difference between humans is not sufficient to explain all these differences in traits that we have. It explains some of them, but not all of them. And so what they termed to explain the rest of the changes is called epigenetics. And that's what I study primarily, genetics and epigenetics. And it's a portion of genetics where it's heritable still, but these are differences in gene activity that don't require a change in your DNA sequence, like I showed you earlier. These are slightly different types of effects or modifications. And so I've just sort of hopefully simply illustrated here what a type of epigenetic modification could be. So in this case, this is DNA methylation. There are other types of epigenetic modifications, but this is one, and one that I study very often. And these epigenetic modifications also control gene expression, okay, whether your gene is on or off. They also control that. And so in this case, we've got a methylated location, which I've shown here over the C. We have 2 methylated locations in little blue man and 2 unmethylated locations in little green man, okay? And what scientists have gone further to show is that, like I said, these methylation marks determine whether the gene is on or off. So you can either have a change in your DNA sequence or you can have a change in one of these epigenetic modifications. Okay? And these both contribute to whether your genes are on or off. And remember that string of genes being on or off, that's our pattern of gene expression that contributes, in turn, to whether we're a little blue man or a green man with orange hair. All right. And so what I wanted to point out here, this figure isn't really, don't study it to detail, but I just wanted to say that these kinds of marks are established early on and they're maintained. But they go through 2 major waves of drastic changes during development. And what's important about this is that these are considered windows of susceptibility. So as I mentioned before, you can have effects that occur early on that affect you later on in life, that lead to disease outcome or healthy, fit outcome later on in life. But these can occur early on. And these are some of the windows of susceptibility because there are changes in these epigenetic modifications at this stage. And so I'm specifically looking at these stages, these windows of susceptibility during development, and trying to understand how does our cell know to have a methylation mark there versus a non-methylation mark? How does our cell understand how to turn that gene on or off? How direct is that correlation between whether it's methylated and it's on or it's unmethylated and it's off? And so the way this works and how epigenetic changes have been proposed to contribute to disease, and actually studies have shown this is true, is that during development you establish your epigenetic patterns, right? And as I mentioned, these contribute to gene expression patterns. And this leads to development of different cells or tissues or organs. So this is how we get a skin cell that's different from our liver cell, that's different from our colon cell, that's different from our hair cell, because of some of these epigenetic patterns. Because every cell in our body has the same exact DNA to some degree. Now what happens is that if you get dysregulation or disruption of these epigenetic patterns at any stage in development, and it is disease causative, they could cause a change in gene expression patterns, which could in turn lead to abnormal development of cells, and that leads to disease. So this is how you end up with certain diseases that have been linked to epigenetic changes, including cancer, which is probably the most well-known and widespread of these diseases. So my question is, what causes these epigenetic defects? This is where I'm really interested. What kind of external effects could change our epigenetic pattern, to change our gene expression, to change our traits? And so there's a number of things, and I hope you can see them on the bigger screen because they're small on mine, but there's a number of things that scientists have shown affect our epigenetic patterns. One of them are environmental toxicants. Air pollution, heavy metals, endocrine disrupting compounds such as bisphenol A, The other thing is our drugs. So cocaine, nicotine, alcohol, these are all considered drugs to some degree. And finally, there's been studies that show that diet affect your epigenome. And this is where I became really interested in diet, because a growing number of dietary factors have been shown to affect our epigenetic states, including methyl donor nutrients, which a lot of people at the NRI actually study, and phytoestrogens, such as those found in soy. And so what I'm trying to understand is how epigenetic marks or modifications respond to diet. Is diet capable of turning these things on or off? How does it turn them on and off? When does it turn them on or off? And how does this affect our health and fitness outcome? And so I just wanted to show you a couple of studies in humans that have looked at potential epigenetic marks, but really they're just diet and heredity, how diet is related to heredity. Okay, and so you don't have to study this figure that I'm showing here, but I just want to say that what they showed in this study, looking at multiple generations of males and females, was that there was an effect that was transmitted specifically through the paternal germline. So remember what I said earlier, like sometimes males and what they eat could also affect the offspring. This is one of those cases. And they found a correlation between the paternal grandfather's diet, so for example, my father's father, and his food intake, whether he had abundant supplies of food or very low supplies of food at a certain time in his life, and how that affected his grandson's mortality. And they found a direct link between the risk of mortality in his grandson and the paternal grandfather. They also found a link between the risk of mortality in the granddaughter through the paternal grandmother. So it's still through the father, but one side is coming through the grandfather to the grandson, and the other side is coming through the grandmother to the granddaughter. And finally, they looked at another example where they saw paternal smoking earlier than 11 years was associated with increased risk of BMI. So there's a lot of studies. These are actually very, this specific study is not so common, but there are a lot of studies out there looking at how diet, potentially through multiple generations, could affect the future offspring. And I just had a second study here because I think obesity is a lot of people are interested in the effects of obesity and how heredity could play a role. And so in this study, they found that, again, don't study the graph too much unless you're just interested in data. But what I wanted to point out with this study was that they found that paternal BMI was linked to decreased DNA methylation at a particular gene in cord blood, which is post-birth. And so in this case, their conclusion or implication of this study was that an obese father is likely to transmit changes in epigenetic modification, specifically DNA methylation, which is what I study, to his son, versus a non-obese father. And so this is an example of potential effects of obesity, heritable effects of obesity. This is very recent. It actually just came out, I think, this week. And so human studies are great, right? But there's a couple of challenges to human studies. One of them is you have limited access to multiple generations. Even in the two studies I just mentioned, they note in their paper that they couldn't collect as many samples as they wanted from as many people as they wanted within each generation, because it's hard to track down people. And people move, people pass away, other things happen. The second thing is that cell and tissue sample collection can be difficult depending on what type of tissue you need to do your studies, right? Who here wants to give me a piece of their liver? No takers. Okay, Brandy wants to give me a piece of her liver. Okay, nobody wants to give out a piece of their liver, right? So most of our cells and tissues are not expendable. Developmental timing is hard to track down. It's hard to organize your studies so they're all at the same timing. That's difficult. Phenotypic variability, what I mentioned, different people responding differently in different disease outcomes, that can pose a noise problem in analyzing your data. Multiple or aggregate exposures is also an issue sometimes because as humans, depending on when you study us, we're exposed to so many things throughout our lifetime: diet, toxicants, behavior, what have you. It just depends on what you're studying, but there's some impact of other exposures that make it really hard to extract the data that we want to study. And the final thing with human studies is that we can make a lot of associations in human studies that are very interesting and very important and necessary, But sometimes it's really hard to say that that causes that. So in the previous two studies, I said, well, grandpaternal food intake affected mortality risk, or is associated with mortality risk is actually what I said, because I can't say that it causes mortality risk because it's just an association. The same thing with the obesity. Paternal obesity is associated with increased risk for decreases in DNA methylation. That's an association. That doesn't mean that if you're obese, an obese father, your child is automatically going to have decreases or changes in his DNA methylation. It's an association. And so I primarily study mice. I study mouse models, mostly because of these reasons that I've listed here. They have a very fast generation time. Inbred strains, you can kind of choose whether your strains are genetically identical, which is kind of hard to do with humans. You'd have to find lots of monozygotic twins, and even them are slightly different genetically. Or you can pick ones that are genetically diverse. So you have sort of your cream of the crop. You can pick whatever you want to study. They also have a lot higher genetic differences compared to humans. So you can compare a lot of differences in how they affect things all at once. You have access to any tissue or cell type you want. You have access to any developmental window that you want to some degree. You can do controlled exposure, right? Because these mice are very well taken care of. They're bred in-house. They're in very nice, clean cages. You can control what they're exposed to. The other thing that's important about mice, obviously, if I want to take these studies and compare them to humans, is that they have a lot of conserved genes between mouse and humans and a lot of conserved pathways between mouse and humans. So the studies that I do in the mouse can many times easily be taken into the human, and I think that's definitely what people actually do. They do their studies in the mouse and translate them to human studies. So that's the goal. And finally, it's an inducible system. So like I said before, with human studies, you can sort of say it's an association, but with mouse studies, you can actually many times test the causality of a change. So if I want to say this change in DNA methylation caused this change in gene expression, I can test that directly in the mouse. Okay? And so for these reasons, I study mice, but like I said, many of these studies have to eventually, if not already, have to be taken into human studies in order to really say this is what is affecting us and this is what's causing or leading to potential health or fitness outcomes. All right. So I wanted to show a couple of mouse studies that are going to be kind of similar to what I'll tell you about my work. And my work, I'm going to give you sort of a preview of what I'm going to do because I've only been here a month, right? So this is a study that they did looking at mouse coat color, so the color of the mouse fur, in response to diet. And so in this particular case, this gene, the agouti gene, controls the color of the mouse fur. And whether that gene is on or off depends, again, on DNA methylation. So when it's fully methylated, the mice are called what we call agouti, or they're sort of a deep shade of brown. It's actually a really pretty color. When it's fully unmethylated, these mice are more of a yellowish color. And so what they wanted to see was whether feeding these mice a methyl donor-rich diet, and I'll tell you a little bit about these nutrients in a little bit. What they wanted to see was if they fed these mice a specific diet that was rich in methyl donors, and I've listed, I've listed some of the foods that you can find these nutrients in. Would that affect their epigenetic states, and would that affect their coat color in return? So this is definitely trying to trace from diet to DNA methylation changes to gene expression changes that would cause their coat color to change. And these methyl donor nutrients are, I think, very cool because they feed directly into the pathway in one way or another in a very complicated metabolic pathway that I chose not to bother you with today. But in a very complex metabolic pathway, they feed directly into the pathway that is responsible for DNA methylation. Okay? So these components or these nutrients are actually metabolized into products that are used for DNA methylation, if that makes sense. And so what they found was what they expected, which was that feeding these mice methyl donor supplements does affect their coat color, and it did affect their DNA methylation. And so this is sort of the classic study of, whether these are sort of direct effects in order, the way that I told you. Changes in diet affecting changes in your epigenetic states, affecting gene expression and traits, which is coat color. In a separate study, which is, I'm bringing this up because I also work with environmental toxicants and how those interact with our diet to some degree to affect our epigenetic states and to affect our gene expression and phenotypic outcome. And in this study, this is the same group actually, and they already knew that this particular methyl donor diet could cause a gain of DNA methylation if you supplement pregnant mothers with it. And these are mice, okay? And what they also knew was that bisphenol A, which I've shown here, which as I mentioned before is an endocrine disrupting compound. So bisphenol A does the opposite. It's an environmental toxicant. It's found in lots of plastics. It's found in receipts. It's just really, really widespread in the United States at least. And what they know was that bisphenol A causes a decrease in DNA methylation. So you have a dietary factor that causes an increase in DNA methylation and changes in gene expression. And now you have an environmental toxicant that causes a decrease in DNA methylation and changes in gene expression. And so what they asked was, if we treat these mice with bisphenol A, which should decrease their DNA methylation, can we fix that effect or rescue it by feeding them this supplement of methyl donor nutrients? And I'm just showing here with this graph that that's what they saw. So if you look at the gray and the white bars, one of those is a control group that was fed just normal diet. The other one are these bisphenol A treated but methyl donor treated mice. So they've got both the environmental toxicant and now this potentially rescuing effect of the nutrition of the diet, the methyl donor diet. And so what this implies is that dietary factors can actually counteract disease-causative toxicants. And like I said, this is a study done in mice. Please don't go out and eat a whole bunch of these things and then expose yourself to some bisphenol A. I'm not saying that that's true. Again, this is causal. These studies have to be repeated in human studies before we start going out and taking lots and lots of things or exposing ourselves to things thinking it's safe because we have a way to fix it. But this is really exciting because it suggests that these toxicants that are very widespread, we could potentially counteract their effects through diet, which is a very easy way to counteract it versus medicine or, I don't know, or operations, whatever you want to choose. And so based on some of these studies that I just showed you and many, many, many more studies that I just don't have time to talk to you about tonight, scientists have come up with a number of questions. And a lot of these questions are actually my questions, but I think we all kind of have the same questions. And so the question really is, what is kind of the effective dosage, the timing, and the extent of effects? So how much of this methyl donor nutrient do I have to take to see this effect, right? When does this effect occur? Is it really occurring early on in development and affecting me later on? Or is it occurring, later on and affecting me later on? And then what's the extent of the effect? Is this happening every time? And that also ties into the frequency of effects, right? So is every person that takes this supplement going to see the same effect? Is every person that's exposed to this toxicant going to see the same effect? And those are questions that we're still sort of narrowing down in the field. And we know that that's actually not true, that every person that takes the supplement and is exposed to the toxicant doesn't have the same effect. And that's where genetics comes in and many other things. And so the other question is, are there specific genes that are affected? Are genes associated with obesity more affected than genes associated with infertility? And so those are the kind of questions that we want to ask. Is it DNA sequence dependent? Is there a susceptibility or resistance to it? And that's kind of what I was saying before. Every person isn't going to be affected the same way by the same type of intake or exposure. So is there something in our genetic makeup that decides how our cell is going to respond to these external factors? The other thing is, what is the role in disease, development, or reproduction? And that's kind of what I said before, are specific genes affected? Are these things playing an equal role in development? I think many people have heard stories, or at least heard of one story, where people are saying certain things are more prevalent now in today's society. Cancer is maybe more prevalent, asthma is more prevalent. And there's always people who say, no, it's not more prevalent, it's just detected more often. But these diseases are seemingly more prevalent in our society. So what's causing that? Is it something like this? Is it a dietary thing? Is it a toxicant, exposure to toxicants? And so that's where these studies will come in. The other question that I think is really important is, are these transgenerational? So I showed you some studies earlier where there were transgenerational effects, right? And the thing that's important there is because, you know, we're going to live our lives and we're going to do whatever and we're going to go somewhere else, right? So we will have an end to this life eventually. But are we affecting future generations? Is this something that we're passing on, you know, and that we should be aware of? Because I don't want to expose my children to something that's harmful to them and their children's children and their children's children. And that also will contribute to increasing prevalence in future generations, right? If we're all passing on a little bit each time, it'll just add up. And so that's a really important question in the field right now. The other thing is aggregate effects. As I mentioned before, we often think of these things very separately. You know, does diet affect our health? Does environmental toxicants affect our health? Does behavior affect our health? But a lot of times we really have to think about a human situation where we're exposed to many different things at the same time. And so that's one of the things that I'm looking into as to whether diet and environmental toxicants, whether that combined aggregate effect either has a greater effect or lesser effect on our health. And that's what I have on there. So I'm going to talk to you just very briefly in the last few minutes about some of the research that I'm specifically doing or planning to do in the lab. And that's why it says ongoing research. And so the first question that I've posed here is, does methyl donor nutrient deficiency during fetal development affect epigenetic reprogramming? So epigenetic reprogramming, like I said, is this establishment of DNA methylation marks. Is one example. So establishment of DNA methylation marks, is that affected by methyl donor nutrient deficiency? And so this is kind of how, it's just a write-up of what I'm going to do in this study, but the important thing about these studies is that they'll address questions like, are epigenetic changes occurring at disease-relevant genes, as I mentioned earlier? So I'm going to look at different genes in the genome and see if they're affected by these changes in diet. The other question that it will address is, how early do they occur? Remember, I said it's an idea of, are they occurring earlier on and affecting us later in development, or are they occurring at the time that the disease occurs? The other question is, do they affect multiple genes, and how? Is this sort of a common mechanism that just one thing has gone wrong in our cell and now our whole genome is affected? Or is it very specifically targeted to a single gene? And finally, are these defects heritable over multiple generations? And I just kind of explain why that's important. And so these kinds of studies will ask those kinds of questions. The second major aim that I'm going to ask is, do genetic differences affect susceptibility to the effects of methyl donor nutrients specifically? And so this is where I was telling you earlier that you could have like a two-hit possibility where one is you have this genetic change or genetic difference that makes you more susceptible to changes in DNA methylation. And then now you have this nutrient that causes changes in DNA methylation. So if I have a G versus a T in this case, which corresponds to a C and an A, if I have this one genetic, one type of sequence at this location versus the other type of sequence, is that going to affect how this diet affects my epigenetic state. And so what I'll do there, and this is just an example like I was saying before, will having a G at that location mean I'm fine and this region gets methylated normally, my gene gets turned on, I'm happy, I'm healthy, I'm fine? Or will changing it to a T, which is going to be in the next person, or really the next mouse, which is what I'm testing, if having a T will now cause this particular region not to get methylated properly, and now my gene is turned off, and I'm not fine. And so that's what this question will test. And it's kind of a very similar scheme, except in this case, I'm going to be looking specifically at animals, or mice rather, that have either induced genetic mutations that I've already shown to cause a change in gene expression, or I've predicted that they could possibly cause this type of change because of what they do. And the other thing is looking at these different inbred mouse strains that have naturally occurring genetic variation. And so these are both relevant to humans in different ways, because some humans do have drastic genetic mutations that cause them to be more susceptible to epigenetic change or to just changes in general. And there's also this naturally occurring genetic variation, like I said, that 0.1 or 0.01% of genetic differences that occurs naturally. And we're perfectly healthy without exposure, but maybe once we get exposed, there's a different thing that happens. Okay, and the question that this will really be asking, is are treated mice with genetic differences more or less susceptible to epigenetic change? And then my final question that I'll be addressing is, does exposure to environmental toxicants alter the effects of methyl donor nutrients? Or vice versa, does the exposure to methyl donor nutrients alter the effects of environmental toxicants? And so this is a similar question to what was asked in the first study that I showed you with mice, where they did bisphenol A and treated them with a supplement of methyl donor nutrients. But in this case, I'll actually be looking at more human-relevant genes. So that study was looking at coat color genes. We don't necessarily have a coat, although we have different colored hair, but we don't have an agouti locus that I know of. So my study will look at more disease-relevant genes. And so the toxicant that I've been studying is called vinclozolin. It's a pesticide. It's sprayed on a very limited number of edible foods and non-edible vegetation. So it's most well known for being sprayed on wine grapes, canola plants, and turf grass. So if you're a guy and you play golf, just don't roll around in the grass or anything like that. But otherwise, there's not really a high exposure, so don't be too worried. But it has similar effects to other endocrine disruptors that we are highly exposed to. But I chose this particular toxicant to study because it is an endocrine disrupting compound. It has fertility defects. Particularly in the male, it affects sperm count and other fertility aspects that I won't talk about today. It's a suspected carcinogen, but interestingly, and most importantly for the types of studies that I do, it affects DNA methylation. And it's been shown to affect DNA methylation over multiple generations. And so I wanted to test this particular toxicant in my studies to look at the timing of when these effects occur. And so I've done some preliminary analysis before I got to the NRI, just to show that when I treat pregnant females with vinclozolin alone, and this is just repeating what's been done already, I'm not finding necessarily anything brand new, but this was just a repeated experiment to confirm that I do have effects on DNA methylation in my mice. And I do see that. I actually saw a 17% reduction in DNA methylation at age 19, which I won't get into what the gene does, but it is a developmentally relevant gene. And defects in its expression in DNA methylation have been shown to contribute to human disorders and disease, including growth disorders, some types of cancer, breast, prostate, lung cancer. So in the mouse, I have seen these types of defects. And so knowing this, what I am planning to do with these guys is do a little bit of aggregate exposure. So these mice that I've already shown have an effect when they're treated with vinclozolin, I will treat them either with a diet supplemented with these methyl donor nutrients or deficient in the methyl donor nutrients. And the questions that I'm going to ask here are these defects observed, that I observed in vinclozolin, the 17% change in DNA methylation. Is it rescued? So is it fixed by exposing them to the methyl donor nutrients, the way that the other study saw it being fixed when they were treated with BPA, bisphenol A? And so that's one question. The other question is it increased? So does a methyl deficiency make it worse? And so these are, like I said, directly relevant to human studies in some ways because we are exposed to lots of toxicants in different ways and forms and times in our lives. And it's a question of, is there an easy fix? Not really easy, but is there something that is very, very widely accessible, such as diet? As I said, these methyl donor nutrients are found in a variety of very common foods in the Western countries, at least. Actually, all countries, I think, because beans are on there, eggs are on there. Most people eat those different things across the world. So it's sort of globally available foods that can be used for this kind of thing. And so those are really the questions that I'm going to ask. I had one final slide, which I might have already covered in my, just going off on a tangent. But I really wanted to sort of hammer the point home about what these kinds of studies address, because I'm doing these studies, there are other scientists doing these studies, and I think it's just really important as scientists to tell our communities what we're doing, how we're doing it, and what is going to be the outcome. What are they going to get out of it? And so really, what is the role of nutrition in determining epigenetic marks? That's one of the questions. How much are they affected, and can these effects contribute to disease? Because if they can't contribute to disease, there's not really a lot of point studying it, unless there's a profitable benefit from using them, so you can have increased health or increased fitness, like maybe athletes or something like that. So the other question is, can detrimental effects of nutrition be inherited? If so, how? And I think there's a lot of studies that suggest it can be and that show it can be, but are these really disease relevant? Do individuals respond differently to nutrition because of genetic differences? I mentioned that over and over again. Can we use this to predict susceptibility in the field of medicine? Can we use it to predict susceptibility? Can we use this to predict treatments or things to avoid? If you already have a genetic mark that makes you more susceptible to the effects of methyl donor nutrients, stay away from them, that kind of thing. And finally, does exposure to multiple, I've got two multiples. Does exposure to multiple factors, such as nutrition and toxicants, increase the risk of developing epigenetic changes? And that's, again, is it sort of an aggregate exposure thing? Can nutrition be used as a reliable way of counteracting effects of our environment? And can we predict potential aggregate interactions or aggregate exposures to avoid? And so I think that's all I had tonight. I just really want to thank you all for your participation by not falling asleep. I've been trying to check out everyone. I haven't seen anyone sleeping yet. But I just want to thank you for coming tonight. We really appreciate having you visit us at the NRI, and I really appreciate you sitting and listening to my talk. Like I said, I've only been here 1 month, and it's just fantastic to have an audience that's interested in anything I have to say. And finally, I have one more slide. These studies cost money, like all studies. And if you're interested in this work, if you think this work is relevant to you or someone you know, and you're interested in contributing, there's a really easy way to do it. And the website's on there if you're interested. With that, I'll take any questions. Yes.
I noticed on the first slide where we were comparing coat colors based on diet that that the yellowest mouse was also the heaviest mouse and the brownest was the thinnest. Can you comment?
Yes, that is a very, very keen observation. She's got a good eye. So, so these studies, so the question that she asked, or rather the comment that she made, was that in the study where they were looking at the effects of methyl donor nutrients, the mouse that was most affected based on coat color was also the chubbiest, and the mouse that was least the mouse that was least affected based on coat color was the chubbiest, and the mouse that was most affected based on coat color was the leanest or the smallest. So there are some studies that suggest, particularly, I'm pretty sure it's the effects of this locus, that they could play a role in weight, in obesity. I'm not 100% familiar with all the things that go on at this locus, but there are some studies that suggest that. Yeah. Any other questions? Or have I stunned everyone to silence? Yes?
How long do you study those mice?
It's a good question. It's a really good question. So she asked, how long do I study the mice? And it depends on the study. And it depends on the type of tissue that I need. So like I said, a lot of what I do, it's very important to have specific stages of development, to look at specific targeted stages of development. And so for many of them, I treat the females during pregnancy, and then their offspring, I study them at multiple developmental stages, either at birth, at 3 weeks, which is when we tend to wean mice, so that's considered sort of adolescence, or adulthood, which is at about 8 weeks, 7 to 8 weeks. And so those are the timings that I study the mice. Those are the timings that the mice are treated and survive for. But I look at them at particular endpoints. So I'm not observing them throughout 8 weeks. I'm just taking them at the end and saying, does what happened to you during your fetal development affect you at 8 weeks of age? Yes.
Well, they always study mice. How long does the mice live? The mouse?
That's another excellent question, and it varies a lot depending on the strain of mouse. Oh, sorry. So she asked me, how long does the mouse live when we're studying them or when we're not studying them? It depends on the strain of mouse. Mice in the lab, I think, live a lot longer than mice in the wild, but I could be wrong on that. I'm just assume that because they're kind of coddled in the lab a little bit unless you're doing types of experiments. My types of experiments, they're well taken care of. And so my mice have lived 2.5 years. That's like the longest I've had them live, but generally they live about 1.5 to 2 years. Yeah, that's what I've seen with most of my mouse strains. That's the range, 1.5 to 2.5 years. Yeah. Any other questions? Yes.
So I had a comment and a question in response to Jessica's question about
The fat and lean.
the heavier mice that are different color.
I get a microphone. Okay. So to respond to Jessica's comment also about the coat color and obesity, I believe that locus has also been associated with cancer. So it's really interesting because we think now that there's a strong link between obesity and different types of cancer.
I did not know that. The AVY locus.
Yes, the agouti locus.
Interesting.
And then the thing I wanted to ask about, I've always been interested in how the parents' habits affect the children, because I think that's kind of a novel thing that people usually think they can just do what they want, and then when they get pregnant or they decide to have kids, then they can straighten up.
That's very true.
So with the study, you talked about the paternal obesity and the changes in methylation. Do you know, or has anybody figured out whether the father's habits change the methylation in adulthood, or is this something that started in development for the father, and then because he ate really badly and drank the Jim Beam in his coffee, that was a second hit, and then that maybe led to something that transmitted to the child? Am I making any sense?
You're making good sense. So you're asking me, if I'm understanding you correctly, whether anyone has looked at the timing of occurrence in the father, even though it's transmitted to the child and to the grandchild. Has anyone looked at when this effect potentially occurred in the father? I think that's a really interesting study to do, but I think it's a really hard study to do because you can do these studies in one of two ways. The first way is to take a man who hasn't become a father and follow him through 3 generations, which, depending on when this man has children, if a minimum of 20 years between each generation or something like that these days, that's 60 years of a scientist's life that you're hanging around waiting for a child to be born that you can study, right? So I think most people aren't necessarily doing it that way. The other way, which is most often done, is you're looking at the grandchild and going backwards through generations. I have a grandchild with an effect. Let me look at your father. Okay, this person has the effect. Let me look at your father. And that, in that way, the grandfather might be 80 years old. Maybe. I'm just estimating here. And that study would be really hard to do. But I think it could be done if you have someone who remembers all of that stuff from 60 to 80 years ago and is willing to give their information to you. So you have to have enough people to do those kinds of studies. But I honestly can't think of a specific study that was done that way, just because of the difficulty.
Thank you.
But that's something that could be done in mice. So, yes?
How long does the nutrients make a change in terms of methylation in the whether it's grown-up or children?
That's a really good question, and one I absolutely do not know the answer to. I don't even know if that's been tested directly, right? Because what would you have to do to ask that question? You would have to feed the nutrients and take the mice at different stages, right?
How about humans?
Oh, in humans. Oh, sorry. So the question was, how long does it take the nutrients to have an effect on epigenetic marks? So example, DNA methylation. So like if I feed someone for 1 week with the nutrients, can I see an effect after that? Is that what you're asking?
Yeah, maybe 1 month, 1 year, maybe 1 month.
I honestly don't know. I think it's a very good question. Do you know? Okay. Yeah, I honestly don't know. I think that it's a very good question. I think that it's, in humans, it would depend because I think your access to tissues in humans is limited. And so just because you don't see an epigenetic change, yeah, oh, I thought you asked me in humans.
You can, yeah, you can get blood cells.
You can get blood cells, but what I'm saying is you can get blood cells in humans, but I guess if you're thinking about the case with the maternal cord blood sample, the study that I was talking about, so you can get blood cells, but what I'm saying is that just because you don't see an epigenetic change in that particular cell type doesn't mean that there's not an epigenetic change occurring due to the nutrients. And so in mice, it would be easier to do because you can look at multiple tissues or cell types and say, well, I don't see an epigenetic change at any stage at 1 week. I don't see an epigenetic change at any stage at 1 year. I see an epigenetic change at 1.5 years, you know? But in humans, I think it's a lot harder to do. So you either see a change or you don't see a change. But I don't think anyone's really, to my knowledge, and if anyone in the audience knows differently, please correct me. But to my knowledge, I don't think anyone's sort of systematically done that study yet in a way that's accurate and relevant. Yes.
Thank you so much.
Thank you.